Hello, my name is Erlyn and these are my friends Sheryn, Hanun, Tasya, and Sherly. Today we will be discussing about a journal titled Triggering the Stringent Response Enhances Synthetic Methanol Utilization in E. coli. First, we will be talking about the background, followed by the methodology, results, discussion, and conclusion, as well as recommendation. Background. Researchers have been gaining interest in utilizing methane as an industrial feedstock in order to reserve natural gas as well as increase biogas generation. In order to harness methane and methanol, native methylotroph is used. However, there are problems with native methylotroph. Firstly, they have slow kinetic growth, and secondly, they lack of established genetic engineering tool. This causes the obstruction of industrial application. As a solution to the problem, researchers engineered synthetic methylotroph through E. coli as a host organism. Why E. coli? This is because naturally E. coli cannot utilize both methane and methanol, but with some genetic modification, they will be able to do so. The picture shows the most common synthetic methanol utilization pathway in E. coli. The green represents necessary enzyme for the pathway, MTH, HPS, and PHI. However, achieving autonomous synthetic methanolotroph is still difficult. The possible reason includes poor enzyme kinetic of MTH, lack of established methanol-dependent growth, improper gene regulation response to methanol, and the incomplete synthesis of proteogenic amino acids from methanol-derived carbon. After more research, it has been said that the PP, GPP overexpression of DKSA and RPOS are able to activate the stringent response that will greatly enhance synthetic methanol utilization. My friends will further explain about the methods on how to achieve this as well as the result and discussion on the analysis.
Now we will be talking about the methods of this paper. The first one is the materials, which are divided into two sub-chapters. The first one is the chemicals, and the second one is the media growth and condition. As for the chemicals, the two worth mentioning ones are C13 methanol, which will be used to identify matter of methanol, and the E. coli NEB5-alpha, which is used for plasmid construction and propagation. In regards to the media growth and condition, it uses lysogeny broth, and for the methanol supplemented cultures, the C13 methanol was added to the final concentration of 60 millimolar methanol, and the C13 methanol is added to be able to identify the matter of methanol. Now, moving from the materials, we will now be discussing about the means of analysis. In this paper, there are three means of analysis. The first one is regarding the strains and plasmids. The second one is RNA extraction, and the third one is just mentioning about all the analytical methods that will be used. In regards to the strains and plasmids, additional from the previous strain mentioned in the chemical section previously, there are two additional strains which are used for the observation of methanol utilization and growth. And what will also be observed as well is the deletion of DKSA and RPOS genes, deletion of FRMA and LRP genes, as well as the overexpression of RPOS and DKSA. Now, in regards to the RNA extraction itself, cDNA synthesized from the extracted RNA was used for transcriptional analysis, and RNA extraction is done to understand the transcriptional exchange of the genes to validify the findings. Lastly, regarding the analytical methods, there are three different sub-methods. First one is biomass concentration, which would quantify the final results and how the engineered E. coli has gathered and assimilate the methanol. The second one is liquid chromatography for methanol, which would just analyze how many methanol is used. And the last one is gas chromatography for C13 methanol labeling, which again is used to see how much methanol has been used.
Next up is the results. There are several trials to improve synthetic methanol utilization in E. coli. The first one is to test three strains which are the parental or wild-type E. coli, PPGPP overproducing E. coli, and PPGPP deficient E. coli in M9 medium with yeast extract 1 gram per liter. As shown in the graph, PPGPP overproducing E. coli or PPGPP plus showed higher methanol consumption than wild-type E. coli, and PPGPP minus or deficient showed a lower methanol consumption. PPGPP is a signal that is involved in stringent response. It causes the upregulation of many other genes involved in stress response, such as the genes for amino acid uptake. On the second experiment, yeast extract was decreased to 0.5 gram per liter, and the results showed an increase on methanol consumption. Because the medium becomes more limited due to reduced yeast, and methanol represents higher percentage in the medium, which causes E. coli PPGPP plus possesses the ability to utilize methanol more efficiently. Transcriptional analysis of PPGPP plus also reveals that amino acid synthesis also plays a role on increasing the methanol utilization. As I mentioned before that, PPGPP leads to upregulation of several biosynthetic pathways. To validate this, PPGPP upregulation was analyzed by detecting the THR-ABC upron, which is known to be upregulated by PPGPP. The results showed that THR-ABC produces threonine, which confirms that amino acid synthesis is also responsible for methanol consumption. Thus, amino acid synthesized throughout 24 until 48 hours, including the activation of serine, threonine, and glycine, enhances the conversion of methanol carbon into biomass. In addition, gene expression is also observed with PPGPP plus, and it showed an upregulation of RPOS. RPOS is central regulator of the general stress response, and showed downregulation on RPOD, which is responsible to encode RNA polymerase. Hence, this confirms the stringent activation because PPGPP inhibits RNA synthesis.
The next experiment was to improve methanol utilization by the deletion of FRMA and LRP in metallotrophic E. coli PPGPP+. FRMA natively detoxifies formaldehyde to carbon dioxide. After the deletion from PPGPP+, it conserved the formaldehyde for the RUMP pathway, which results in minimizing methanol carbon loss. On the other hand, LRP is natively represses threonine catabolism. After the deletion from PPGPP plus FRMA, it alleviates the repression of threonine catabolism, and shown to mimic the stimulatory effects of threonine catabolism on synthetic methanol utilization. Both deletions resulted in improved 13C labeling of RNA, serine, succinate in the respective metallotrophic E. coli PPGPP+. The last experiment was to trigger the stringent response by the KSA and RPOS overexpression. Here in the figure is the comparison between the deletion and the overexpression of the KSA and RPOS. The result clearly shows that the overexpression of the KSA and RPOS exhibited a significantly higher Tritanti labeling in glycolytic, TCA cycle intermediates, and amino acid. It is proven that regardless of the activator component, the stringent response activation improves the utilization of synthetic methanol in E. coli.
Next, discussion.
This research proved that by examining the PPGPP division and also overproducing metallotropic E. coli strains, the PPGPP overproduction during the metallotropic growth significantly improved the methanol utilization, which can lead to the increase of cell growth. The C labeling analysis confirmed a higher amount in methanol utilization in PPGPP overproducing metallotropic E. coli compared to the PPGPP division and also the parental metallotropic E. coli strains. Importantly, the PPGPP overproduction metallotropic E. coli achieved improved synthesis from methanol carbon of those limiting amino acids that are minimally synthesized from methanol carbon in the control metallotropic E. coli strain. This PPGPP overproducing metallotropic E. coli showed increase in the threonine synthesis from 13C-methanol as the result of direct THR-ABC upregulation, as well as the synthesis of serine from 13C-methanol as a result of indirect SERA and SERC upregulation. Other than that, the overexpression of both TKSA and RPOS also increased the methanol utilization significantly in the same way as the PPGPP overproduction. This suggests that general stringent response activation, regardless of the activated component, is the strategy to increase synthetic metallotropic in E. coli strain.
Conclusion. All analysis done on the strain and plasmid of E. coli in order to produce more natural gas and biogas product of industrial uses in methanol medium results in the activation of stringent response. Thus, it can be seen that PPGPP helps activate stringent response, which in turn improves methanol utilization in E. coli. Recommendation. We believe that a better initial strain that is able to synthesize all proteogenic amino acids from methanol carbon and is able to regulate essential gene expression for growth from endogenous conditions resulting from the regulatory responses of the organism to the unnatural substrate will prove more successful toward realizing autonomous methyl trophy. That's it from us. Thank you.